Hello everyone, my name is Zebang Chen. Today I would like to share a paper named In Vitro Prototyping of Limonene Biosynthesis using Cell-Free Protein Synthesis, which is published on the Metabolic Engineering. And this research team behind this article is Professor Jewitt's group at Northwestern University. First, I would like to introduce the background of this research. The isoprenoid are a diverse class of over 40,000 known compounds with applications in various fields, including the pharmaceuticals, fragrance and flavors, insecticides, chemical precursor, and biofuels. While some isoprenoids are already produced at a commercial scale, engineering microbial strains to produce a novel isoprenoid remains challenging due to three major factors. The first is a long optimization cycle for metabolic pathways and the complex regulation of the heterologous enzyme expression and the metabolic constraint imposed by intracellular regulation. And the CFME, the cell-free metabolic engineering, offers a promising solution to the challenges in metabolic pathway optimization by enabling the rapid adjustment of enzyme concentrations, substrate, and cofactors. This approach facilitates the high throughput screening of the hundreds of different pathway configurations, accelerating the discovery of optimal metabolic routes, and CFME has already been successfully applied to the biosynthesis of fatty acid and demonstrating significant advantages in metabolic engineering and synthetic biology. And the iProbe, the in vitro prototyping and rapid optimization of biosynthetic enzymes. This approach is a novel strategy for optimizing the metabolic pathways. It utilizes the cell-free protein synthesis, the CFPS, to express enzyme in vitro, enabling immediate testing of their catalytic activity without requiring intracellular expression. This method provides a modular approach to assembling different enzyme combinations, bypassing complex regulatory interactions with living cells. And iProbe has been demonstrated to accelerate the optimization of 3-hydroxybutyrate and N-butanol biosynthesis, significantly reducing the optimization cycle from 6 months to just a few weeks. So here are the research objectives of this paper. First is optimization of 9 enzyme pathway using iProbe to find the best combination of enzyme homologous and evaluate the effect of cofactors on enzyme activity and optimize the metabolic fluxes and improve the limonene yield and shorten the optimization cycle by iterative optimization strategy. And then to validate the modularity and scalability of the method and test whether it can be applied to the biosynthesis of other terpenoids. So next, I will describe the materials and method used in this paper. Here is the information structured into 3 sections: the strain, plasmid, and lactate preparation. For strains, so the E. coli BL21 strain was used for cell-free protein synthesis. The cells were cultured in 1 liter of 2 times YTPG medium, and at OD 600 equal to 0.6, 0.1 mile more IPTG was edited to induce a T7 RNA polymerase expression. The culture was continued until the OD600 equal to 3.0, at which point the cells were harvested by centrifugation for further processing. And for the plasmid, the target genes was cloned into the PGL1 plasmid, and its sequence was amplified using a PCR with specific primers. The forward primers was designed to encode the sequence after the second condom of the target genes, and the reverse primer encodes the sequence before the stop condom. To improve the cell-free protein expression efficiency, an N-terminal expression tag was introduced for enzyme homology selection. Sequences corresponding to relevant EC number were retrieved from the random database, aligned using the muscle 3.8.425, and used to construct a phylogenetic tree based on the Jukes-Cantor genetic distance model, and then the neighbor joined the method. A total of 45 enzyme homologers were selected and condom optimized for E. coli expression and synthesized by Gene9 or Twist Bioscience. And for the lysate preparation, following the cell harvesting, the cells were washed and centrifuged to remove the preceptors. The resulting S13 cell lysate, the extracts were fractionated and stored at the -18 for subsequent using in cell-free protein synthesis experiment. Next is about the cell-free protein synthesis reaction. The PNOX-SP system was used for this protein synthesis, and it can allow in the rapid expression and testing for biosynthesis enzymes without requiring the intracellular expression. The react system included the 13.3 microliter of S13 lysate in a 50 microliter CFPS reaction with essential components such as nucleotides, cofactors, and 20 standard amino acid. Additional components, including the tRNA mixture and so on, and were added to support the protein synthesis. Since the T7 RNA polymerase expression was already induced by the IPTG, it was not required in the CFPS reaction. Reaction was incubated at 30 degrees Celsius or 16 degrees Celsius for 20 hours, after which the products were frozen in liquid nitrogens and stored at -18 degrees Celsius. This CFPS approach, as a part of iProB strategies, enables a rapid optimization of enzyme concentrations, substrates, and cofactors, accelerating the metabolic pathway development and high-throughput screening for biosynthesis optimization. To qualify the yield of the cell-free protein synthesis reaction, 40C leucine was used to label the synthesized protein. At the end of the reaction, the centrifugation was performed to remove the insoluble proteins, allowing the measurement of the soluble proteins. The protein was then purified by the TCA precipitation and qualified using the liquid counting to measure the radioactivity. Finally, the protein concentration was calculated using the standardized formula. For lemonade synthesis, the reaction was incubated at 30 degrees Celsius, and 30 microliter for dodecaine was added as an overlayer to prevent the volatilization and ensure the stable linear retention during the action. to monitor the limonene production over time. Samples were collected at 3, 4, 5, 6, and 24 hours and analyzed using the GCMS, which provides a precise qualification of volatile organic compounds. Additionally, the HPLC was employed to detect and characterize other metabolites in the reaction, ensuring a comprehensive understanding of the pathway performance, possible byproducts, and potential metabolic bottlenecks. This analytical approach allows for accurate assessment of enzyme efficiency and metabolic flux, contributing to the optimization of the iProB-based biosynthetic strategy. So basically, this figure presents a phylogenetic analysis of the limonene biosynthetic pathway and key enzymes, highlighting the both metabolic road for limonene synthesis and the evolutionary relationships among critical enzymes involved in the pathway. So figure A shows the biosynthetic pathway starting from the glucose and proceeding through the glycolysis to produce acetyl-CoA, which enters the MVA pathway, and the pathway involves several key enzymes, including the ACAT, HMGS, HMGR, MK, PMK, PMD, IDI, GPPS, and LS, and ultimately leading to the production of the limonene. And the figure B provides the phylogenetic trees for 5 key enzymes in the pathway, comparing the homologs from various organisms. This includes the MK, PMK, PMD, IDI, and LS. The tree reveals the evolutionary diversity among the homologs, offering the insights into the enzyme selection for metabolic engineering. This analysis aids in identifying optimal enzyme variants for improving the limonene biosynthesis, a key aspect of the pathway optimization in iProB-based metabolic engineering. It is regarded as a summary diagram of the whole paper. And this part illustrates the application of the iProB to 9 enzyme pathway for producing the limonene, shows the process of enzyme selection, expression, and evaluation of pathway performance. So the figure A shows the workflow of the iProB approach, where enzymes are expressed using the cell-free protein synthesis and assembled into different pathway combination of metabolic testing. The process includes a pre-enriched lysites containing the essential enzymes and CFPS-enriched reactions that enables rapid prototyping of enzyme variants to optimize the limonene biosynthesis. And in figure B, present enzyme expression levels comparing the total protein production and solubility at 13 degrees Celsius and 16 degrees Celsius. Lowering the temperature to a 16 improves the solubilization of the certain enzymes, aligning with a known strategy for enhancing the protein folding in both in vivo and cell-free system. And figure C and figure D, they both show the initial reaction rate and final limonene titer, respectively. For more enzyme variants, screening 51 enzyme pathway combination revealed the substantial variance in enzyme activity and overall limonene production, with specific homologs outperforming others. And figure E introduced the TREE score, and this is a metric combining the final titer, initial rate, enzymes solubility to rank different pathway configurations. This ranking facilitates the rapid identification of optimal enzyme homologous, while also eliminating the low activity or inactivity candidate from further testing. The results show that the CFPS expressed enzymes exhibit the metabolic activity comparable to those from the enriched lysate, validating the effectiveness of the iProbe in pathway optimization. So in this section, they investigate the key roles of cofactor in optimizing the multi-enzyme pathway and their impact on limonene biosynthesis efficiency. First, a comparison between the initial pathway enzyme optimized pathway without the supplemented cofactor. FIGO-A shows that even in absence of the external cofactor, the optimized enzyme set exhibited the higher activity, indicating that the improved enzyme combination itself was more efficient. And in FIGO-B, by systematically testing the different concentration of ATP, NAD+, and CoA, the study found that the higher level of these stuffs increased the TRE scores, particularly in the optimized enzyme set. And they further analyzed revealed the GPPS and PGGPPS exhibited the distinct activity under different confocal conditions. So with PGGPPS displaying the superior activity in optimized cofactor environment compared to the PAGPPS. Finally, examine the combination of the GPPS with various limonene synthesis shows that the PGGPPS, particularly under the optimized cofactor condition, achieves the highest TRE scores, which suggesting that this combination is more favorable for efficient limonene biosynthesis. In summary, this part of study shows the strategy cofactor optimization can significantly enhance multi-enzyme pathway performance, increasing the metabolic flux and providing the systematic experimental data to support the further optimization of cofactor in cell-free metabolic engineering, ultimately facilitating high efficient limonene production. So this part illustrates the iterative screening process for optimizing enzyme homologous under multiple cofactor conditions to enhance limonene biosynthesis. In Figure B, it shows the screening rounds. The study employed a stepwise iterative screening process from round 3 to round 9, progressively refining the enzyme homologous and cofactor conditions. Each round involved testing an increasing number of enzyme cofactor combinations, starting with 24 reactions in round 3 and reaching 120 reactions in round 7, before concluding with 18 optimized reactions in round 9. This approach allowed the identification of high-performing enzyme variants and optimal cofactor conditions. And in the Figure B, it's about the TRE score progressions. TRE scores for each round show that as the screening round progressed, enzyme performance improved and with the higher TRE scores observed in the later round, in round 8 and round 9. So this trend shows the effectiveness of iterative screening in optimizing pathway performance. And in Figure C, it's about the homolog specific performance. This section ranks the best performing enzyme homologous across different pathway steps, including the GPPS, IDI, PMD, PMK, MK, HMGR, HMGS. Some enzymes showed significant improvements, while others had already been optimized in early rounds. These results confirm that selecting the right homologous for each step substantially impacts overall pathway efficiency. And in the last figure, it's about the metabolite and limonene production over time. This graph tracks the lemonade production, which is red, and the metabolic accumulation is black and gray. Over 30 hours, glucose is progressively consumed, leading to the production of the byproducts such as lactic acid, succinic, and ethanol, with the lemonade production steadily increasing. This highlights the effectiveness of the fine optimized enzyme cofactor system in driving the metabolic flux towards the lemonade synthesis. So this part shows how an iterative screening approach successfully refines enzyme homologous selection and optimization to maximize the lemonade production. And by continuously testing and improving the enzyme cofactor combinations, the study identifies the high-performing homologous and optimal metabolic conditions and can lead to the enhanced pathway efficiency and increase production yield. So in this section, we discuss the bioproduction of additional terpenoids, expanding beyond the lemonade to include the alpha-pinene, beta-pinene, and bisabonine. So in the figure A, it shows that the biosynthetic pathway leading to these terpenoids. The GPP serves as a key intermediate that can be converted into lemonade or further extended into the FEP to produce the bisabonine. And in figure B, it qualifies the C10 production at 24 hours, showing that the lemonade produced by the MSLS has highest titer, whereas alpha pinene and beta pinene produced by AGPS and PAPS exhibit the lower concentrations. And in figure C, it evaluates the bisabonine production at 72 hours, shows that AGPS enables the synthesis of bisabonine at a significant higher concentrations at 5 mMol, suggests that its strong biosynthetic potential. So this part of research highlights the flexibility of iProB system in generating the diverse terpenoids with a potential application in biofuels and pharmaceuticals. To sum up, in this study, they optimize the lemonade biosynthesis pathway in a CIPS cell-free system using iProB method and can achieve the following breakthrough. First, the non-enzyme lemonade pathway was optimized and the yield was successfully increased 25-fold. And systematically screening the 580 plus enzyme homologous and cofactor combinations, identify the PGGPS, SIIDI, and MK as optimal enzyme candidates. And the cofactors such as ATP, NAD+, and CoA were optimized, and the dependence of different enzymes on cofactor concentrations was found. And the cell-free method provides a high-throughput screening platform for future metabolic engineering and can be extended to other isoprenoid compounds. That's all. Thanks.